Welcome, the Office of Programs to Enhance Neuroscience Workforce Diversity within the National Institutes of Neurological Disorders and Stroke is pleased to welcome you to the Open Stage monthly webinar series. This is a targeted outreach effort for our diverse neuroscience community to learn about NINDS programs and network with NIH and NINDS program staff. Each webinar will feature a different NINDS program, division, or cluster with the goal of increasing the representation of diverse groups in our portfolio. So this month, we are so excited to have with us NIH program staff who will speak on the IGNITE program. And before we get started, I just have a few housekeeping items. Dr. Spriggs, if you could go to the next slide. All right, awesome. So few meeting reminders. I know we've done a lot of Zoom calls, but all attendees will be initially muted. So please submit your questions using the chat function, and we'll have a Q&A at the end. A reminder that we are recording this meeting and we will have it transcribed and archived on the NINDS website. So if you miss anything, feel free to go back to the recording. Details about where to find that archived version will be posted on social media and our listservs, and I'll also send all registrants an email afterwards with resources and emails of our panelists. So you'll have all of that information as well as the slides. So we'll have that for you. And please do stay connected with us and feel free to email us if you have any questions about the webinar series or any of our other resources. So with that, I'm very excited to pass it off to Dr. Shardell Spriggs to get us started.
Hello, everyone. Thank you, Anna, for the introduction, and thank you again to Open for hosting this webinar for IGNITE. We greatly appreciate the opportunity for collaboration. Today we have from IGNITE, Dr. Becky Ruth, who is the director of the IGNITE program, Ms. Shruti Thomas, who is the health program specialist, Ms. Ashley Givens, the operations coordinator, myself, and Craig Hopp from NCCIH as a special guest. Again, I'm Shardell Spriggs, the program director in the Division of Translational Research at NINDS. And today I will walk you through the IGNITE program. So IGNITE stands for Innovation Grants to Nurture Initial Translational Efforts. The goal of IGNITE is to support early stage therapy development and to help investigators bridge the gap between basic research and more advanced translational research programs. IGNITE is specifically set up as a feeder program for the Blueprint Neurotherapeutic Network and VPN Biologics program. A great way to be successful in this pipeline is to take a look at the funding opportunity announcements, or FOAs, for VPN, and prepare your IGNITE grant with them in mind. Make sure the aims and research in your IGNITE grant will meet the entry criteria outlined in the VPN FOAs. IGNITE consists of a suite of funding opportunity announcements, which are listed here. They have specific goals and scopes of research that we will explain in the next slides. All three FOAs have the same period of support of 3 years and the same budget of $750,000 in direct costs over 3 years, with no more than $499,000 in any given year. The next due dates are June 21st and October 18th. IGNITE FOAs utilize the R61 R33 mechanism. This is a bit different than a traditional R01. The R61 R33 is what we call a phase mechanism. In the first phase, you demonstrate feasibility and get prepared for the R33. You would validate the assay for your high throughput screen or optimize compounds for your proof of concept efficacy study. This phase is where you show feasibility and prove your project is worth the additional investment of the R33 phase. So how do we make that decision of what advances and what doesn't? You will have a set of go, no-go milestones that are driven by clear and quantitative success criteria. These are milestones that you propose and include in your application, and we'll help show you what good milestones look like. If these milestones are met, you will move forward to the R33 phase. We call this the main event. This could be your high throughput screen, proof of concept study, or external validation of your animal model. The research performed in the R33 is usually what your project needs to get you the entry criteria for the next stage program, such as BPN. There is only one go, no-go decision point between the two phases. So if there is only one point within the project, you will need to meet these milestones. You decide how many years you spend in each phase as part of your application are within limits. And that will vary by project and could be based on how much effort it will take you to get ready for the main event. The transition for the R33 phase, that is whether your milestones have been met, is determined via an administrative review by NINDS program officers. So these do not go back to peer review for that decision. The benefit is that this transition can happen quicker and we can go from the prep to the main event without any lapses in funding. So we are able to utilize an administrative review because a review panel would have previously seen and commented on your milestones since you include them in that application. It is imperative that the milestones are clear and quantitative so that there is no question as to whether they have been met and if the project should advance. We use milestones to de-risk translational research projects. With milestones in place, the investigator can propose riskier science and the institute can fund riskier science. Within IGNITE, the second phase is completely dependent on the first phase of the project. And projects like this would probably score poorly in the R01 space, but it's expected here. Reviewers for IGNITE are well aware that program staff have the ability to stop projects that do not meet the important milestone checkpoint, and therefore are comfortable supporting projects that have this built-in interdependency. What makes this all work are milestones. The concept of milestones and how to construct clear and quantitative milestones can be hard for investigators that traditionally operate in the R01 research space. Milestones are not a list of activities that you will do in the project. Milestones should be the bar that you need to meet to prove the project warrants further investment. For a milestone, you would never say that you're going to test 500 compounds to see if they prevent seizures. You would state that you will identify 2 compounds that prevent seizures in a model by reducing your set criteria by a certain percentage. The idea here is that we are not concerned if you have to test 500 compounds or 5 compounds to find those 2 that hit the success criteria. So that's a general idea, but let's go through some examples to demonstrate the differences in weak versus strong milestones. So to scale up a compound is our example of a weak milestone. You need to say you are going to generate a certain amount of the compound, so X grams, with a stated purity as determined by the method of your preference. Another weak milestone is to say you're going to do PK studies and then select compounds with the best characteristics to test in animal models. Well, what characteristics? And what is that threshold that says these are the best? We need to know that. So we have to see the properties you're talking of: half-life, brain to plasma ratio, microsomal stability, for how long and at what temperature. So the criteria should prove that the compound is worth developing, that it's real, that it works, and do not set too low of a bar. This must be an improvement over the preliminary data that has been presented in your application. Another example is to fully optimize and validate an assay. It would be better for you to include the statistics within that assay. For treatment with a protein to decrease seizures compared to control. What kind of treatment is this? So including the route of treatment, the duration, how much of a decrease you want to see, your animal group numbers, the statistics, things like that are all needed in the milestones to make them quantitative. So hopefully you see the differences. Anyone should be able to pick up your milestones, even if your project has been handed to a new PO on the grant, and unambiguously interpret the level that needs to be met for success. This protects you as well as the Institute. And there are more examples of milestones on the Ignite website. So looking at the individual FOAs, the first one is PAR 21124, which is for assay development. The goals here are to develop new assays and then use those assays for screening to identify new agents for therapeutic development. The entry criteria are that they need to be novel, but also have a strong biological rationale, address the relevance for the therapeutic development. This should be an improvement over what's currently available, and provide a rationale for your test agents or library. This can be large or small as long as you justify the use or reason. Some activities you might do in your first phase include the following, but are not limited to these. Development of assays to support a screening funnel for future in vitro or ex vivo efficacy and or potency studies. Evaluate the purity and identity of the therapeutic. Development and optimization of your high throughput screen, or if needed, a combination of assays might also be useful. So you would transition from the R61 to the R33 phase when your assays are fully developed and optimized. This would include any physiochemical characterization of your test compounds or cell line selection development to be used for assay validation. Examples of activities appropriate for the R33 phase include, but are not limited to, the screen you set up, preparation of therapeutic agents, confirmation of structure, any further optimization, assessment of PK parameters, looking at off-target activities as just a few examples. At this time, I'd like to invite Craig to talk about NCCIH Interests in this funding opportunity.
Yes, thank you, Shardell. So NCCIH certainly is interested. We are only participating in this single part of the Ignite program, but we are very interested in looking at analgesic targets. So really all the guidance you were given about this PAR applies to NCCIH Interests. So the design, the goals, the phases are all the same. We're just focusing on a subset of those, which is the target must be for NCCIH to be interested in, for this to be supported by NCCIH, must be analgesic related. And the intervention must be a natural product. And we are actually encouraging use of libraries of natural products for screening purposes. And there are a variety of those out there, either commercial or personal. People have their private collections. We particularly noted that the NCI, the National Cancer Institute, and their natural products branch has developed a very large repository that they are making freely available to anyone. So we have particularly highlighted availability of that, but you're welcome to use any library or source of natural products that you see fit. But again, for this to be assigned to NCCIH, there must be exclusively natural products that you are studying, and it must be focused on an analgesic target. But other than that, all the other parameters around this PAR also apply to NCCIH. So happy to take questions later on if people want to explore further. Thanks.
Thanks, Craig. So there are activities that would make your project non-responsive to PAR 21124. If an application proposes any of these studies, we will withdraw the application without review. So please review these in the FOA and contact us if you have any questions. It is not our goal to throw out projects. We would much rather work with you to develop a responsive application or point you to the appropriate program. Applications that are focused on indications outside of the mission of NINDS or outside NCCIH's interests, as you just heard about, will be considered non-responsive. The Division of Translational Research at NINDS has a number of programs and funding opportunities to address all stages of translational research. It's important that you apply to the appropriate FOA and appropriate program suited to your research stage. In general, things are going to be non-responsive if they're basic science or if they are further developed and better suited for blueprints. There are also biomarker and devices programs that projects may be better suited for. So the development of risk detection, diagnostic, prognostic, predictive, and prevention biomarkers, applications with the primary focus of developing PD biomarkers, development of devices, surgical procedures, diagnostics, and rehabilitation strategies are all non-responsive. Development of assays or probes to support basic understanding of disease for basic research, target identification, studies of disease mechanism. In vivo efficacy studies are supported by a companion IGNITE FOA. Clinical compound manufacture development of bioassays for clinical use. If you're ready for clinical use, you're beyond IGNITE. IND enabling studies are better suited for blueprint neurotherapeutics. And then human subjects research, except for research that meets the exemption 4 criteria. And please contact us if you're unsure about this last point. PAR 21123 is for the development and validation of model systems. The goal of this funding opportunity is to promote a significant improvement in the translational relevance of animal models and ex vivo systems that will be utilized to facilitate the development of neurotherapeutics. We are looking to provide translational tools for the drug discovery and development process that will make a significant difference, not just for your lab, but for the whole disease area, and will be utilized by other investigators or companies. So in addition to having a strong biological and translational rationale for the model, there should also be evidence of value for future drug discovery and development. There are 2 important concepts to define for the context of this funding opportunity: internal and external validation. Internal validation addresses the consistency of the experimental design. It involves characterizing the precision, reliability, analytical sensitivity, accuracy, and dynamic range of the endpoints used to assess the animal model or model system. External validation refers to the extent to which results of a study can be generalized from one setting to another. So here, we are looking at applicability from the model to human disease. There are 3 types of external validation we ask you to address in the FOA: face, construct, and predictive validity to the extent possible. So face validity refers to the similarity between the model and the clinical manifestation of the disease, such as symptoms and morphology. Constructability is the similarity between the model or model system and the physiological basis of the human disorder. And predictability is the probability that a validated therapeutic intervention will have the same effect in the model as in the clinical disease population. Examples of activities for the R61 phase of this funding opportunity include the initial development of the model or ex vivo system. You may develop and validate a model from scratch, or you can take a model that has not been well validated. Other validating activities, including an optimization related to the feasibility, import range, or sensitivity, and internal validation for the endpoints used, as well as any scale if needed. Examples for the R33 phase include all external validation studies, including comparisons of phenotype to human disease, comparisons of disease etiology in preclinical species to what is known about the human disease, and efficacy of clinically validated therapeutic agents in the new model system. So there are some activities that are included non-responsive. That includes developmental models for the purpose of understanding disease etiology, identification of CNS drug targets, discovery of disease initiation, remission, relapse, or progression biomarkers, applications with the primary focus of developing pharmacodynamic biomarkers, aim study at identifying, optimizing, evaluating, or developing a potential therapeutic agent, discovery or development of devices, device drug combinations, surgical procedures, diagnostic or rehabilitation strategies. And again, there are some other programs here at NINDS that some of these are more suited for. The last FOA in our IGNITE suite of opportunities is PAR 21122. This is the Neurotherapeutic Agent Characterization in vivo Efficacy Studies FOA. This FOA provides funding for studies to demonstrate that early stage neurotherapeutics have sufficient biological activity to warrant further development. Therapeutic agents include small molecules, biologics, or biotechnology-derived products. We use a combination of parameters, including target engagement, PK, and in vivo efficacy studies to determine the feasibility of the candidate therapeutic agents. Necessary entry criteria that you should be sure to illustrate in your application include novelty, such as new knowledge around targets, mechanisms, pathways, or therapeutic indications. Projects should aim to develop or repurpose therapeutics that are significant improvements over existing therapeutics to treat neurological disorders. There must be a strong biological rationale for the intended approach, along with preliminary data or literature-based evidence supporting the feasibility and importance of your proposed approach. Preliminary data should be rigorous and from well-designed experiments. The presented evidence within your application should show or support that the therapeutic agent has the potential to be therapeutically viable, and also the robustness of your PD measures and in vivo efficacy models. Projects should include a description of the unmet need for the therapeutic agent and a clear justification for how the findings from these studies are relevant to treatments for disorders that are within the NINDS mission. Projects should explain how the candidate therapeutic will be optimized and advanced towards later stages of discovery once the R33 phase is complete. Some examples of activities for the R61 phase for this FOA include those that are listed here. While this is not an exhaustive list, the next two slides taken together will give you a clear picture of how all of our FOAs fit together. So activities usually found in the first phase are preparation of the therapeutic agent to support proposed activities, characterization of therapeutic agents. This could include, for example, confirmation of purity, stability, PK properties such as in vitro absorption, distribution, metabolism, and excretion. Limited SAR to optimize the candidate therapeutic agent for in vivo efficacy testing, studies to develop dosage forms to support proposed PD measures, PK studies to determine direct or indirect therapeutic agent levels, studies to confirm that therapeutic agents reach and engage the target site directly or indirectly at a level that exceeds in vitro pharmacological potency, studies to confirm selectivity of the therapeutic target, limited studies to identify potential pharmacodynamic biomarkers specific for the purposes of developing the therapeutic agent, and also studies informed to design and refine the PD measures or in vivo efficacy models. At the end of the R61 phase, there are criteria that should be met to transition to the R33 phase. We like to see all necessary preparation and characterization of proposed therapeutic agents, chemical synthesis, scale up to provide material for the proposed project, in vitro or in vivo potency studies, in vitro organ, tissue, or cell culture systems, all PK studies, design, refinement, and validation with pharmacodynamic markers. You should also have a further refined and validated in vivo study design that meets the NINDS rigor guidelines and will allow demonstration of dose and exposure response. Examples of activities for the R33 phase include, but are not limited to, those that are listed here. And so there are some activities that are either not in scope of the IGNITE program or fall within another IGNITE FOA. Some examples of activities that are non-responsive to this FOA are shown here. You've heard some of these before, but it's important that you do not submit applications that are non-responsive or they will be withdrawn. So development of de novo animal models, assay development, discovery of novel therapeutic agents as covered by a companion IGNITE FOA, discovery and development of risk detection, diagnostic, prognostic, predictive, or prevention biomarkers, although use of existing biomarkers is appropriate. Applications with the primary focus of developing pharmacodynamic biomarkers is covered under a different program. GLP toxicity studies, investigational new drug, enabling studies with optimized therapeutic agents are non-responsive. And then again, the discovery or development of devices, device drug combinations, surgical procedures, diagnostic or rehabilitation strategies, again, are non-responsive. And studies of disease mechanism or disease target identification. So really quickly, we'd like to give you some general tips that are applicable for all three FOAs. The big thing is to contact us in advance. We understand that this phase mechanism is a little different than the R01. So the best thing to do is to send us an AIMS page. We can make sure you're at the right institute, in the right program, and are applying to the right FOA. We're very happy to set up a call with investigators when we are all here and available to help you. Rigor. We would like you to make sure you include a rigorous study design. NINDS has issued a specific notice addressing this and it's listed here. This includes not only how the studies are constructed, but how you collect the data, interpret the data, statistics, power the experiments, and even the strength of the preliminary data used to support your scientific premise. You should have a multidisciplinary team. We want you to note the multidisciplinary review of IGNITE projects. The review panels for IGNITE have expertise, not just in the disease, but also in assay development, medicinal chemistry, animal models, and beyond. The panel is comprised of clinicians, industry, as well as academic scientists, all so that you get a good multidisciplinary look at your application. Each application will have 4 or 5 reviewers, and this is on purpose to make sure you get all of these perspectives. The best way to write a strong application that satisfies a multidisciplinary review panel is to have a multidisciplinary team yourself help craft this application. We would like you to strive to increase the diversity of your team. IGNITE has added stronger language to these FOAs to stress the interest in diverse research teams. Research has shown that diverse teams working together and capitalizing on innovative ideas and distinct perspectives outperform homogeneous teams. Scientists from diverse backgrounds and life experiences bring different perspectives, creativity, and individual enterprise to address complex scientific problems. This is an interest across NIH, and again, there have been notices issued for this as well. Please discuss the intellectual property, especially for therapeutics, around your project. You do not need to have patents filed at this point, but you do need to convince reviewers that you're thinking about the future and have a minimum freedom to operate. A letter of support from your tech transfer office saying that they'll work with you is helpful. And also have a therapy development plan. Convince reviewers that this project has a path forward if you are successful and hit all your goals during this 3-year project period. Show you are thinking about the future and how to gather the expertise needed to help prevent or address pitfalls. This could be through collaborators or current expertise you have on your team. if you're a small business, you are eligible to apply to Ignite, but we recommend you consider the small business SBIR program. NIH sets aside funds for this program. And if you are eligible, then you're only competing with other small businesses for this pot of money instead of competing in the larger pool of everyone else. And these last few bullets again address the non-responsiveness of applications. Ignite requires go, no-go milestones. Applications without milestones will be withdrawn. It's important that they are quantitative because reviewers take a look at these and consider them in your score. Please see more examples on our website. Clearly demarcate the R61 activities within your project from the R33 activities. Include this in a timeline in your project. Pay attention to the list of non-responsive activities that we've told you about. They are all listed in the FOA for your reference. And again, pay attention to human subject research designations. And please feel free to reach out to us if you have questions on your specific project. And again, for resubmissions, just going by the numbers, most applications will not get funded. If we get 30 applications in and we fund 4, most will fall within that group of 26. We encourage you to resubmit your application. Resubmissions have the same due dates as new applications. And this year, that's June 21st and October 18th. You should resubmit because resubmissions typically score better than initial submissions. This is due to a number of factors, but including because your new application has the benefit of receiving a written critique from review, where you can respond to these reviewer critiques. You do not have to agree, but it's very good to address them. So please, if you apply to Ignite or have previously applied to Ignite and are interested in resubmitting your application, reach out and contact us once you receive your summary statement, and then we can work with you. So at this time, I will hand it over to Shruti and Becky if we have any questions. If we don't have time to get to your questions today, please feel free to email Becky or any of us on the Ignite staff, and we can make sure we get your questions answered.
Thanks, Shardell. If you have any questions, please list them in the chat and we can go through them one by one. And then for any project specific questions, you should reach out to us and we can try with a specific aims page and set up a call. So the first question is, what are the opportunities or barriers to applying overseas? What does applying with international collaboration look like?
So for overseas applications, you have to justify that. And so there are 2 ways NIH funds overseas applications. You can be an overseas application fully if you're the PI, or you can just have a foreign component in the application. The foreign component within the application has a lower bar, but they both have to be fully justified as to why the research should be done outside of the United States. And I don't know if you want to add to that, Becky.
Nope, you got that right. Thank you, Shardell.
The next question is, can PAR 21-124 be used to optimize or improve existing assay methods?
If that one's mine, I'll say yes, it can be.
Can PAR 21-124 make use of CROs to provide custom libraries?
Again, yes, there's no problem with that.
This is Craig. I guess I'll just add that I agree, there's no problem with that, but you should still be able to, in your application, describe what those libraries are going to be. Saying I'm just going to have a CRO provide me a library is probably not going to fly very well in an application during review. You'd have to be able to describe what exactly is it they are going to be providing in terms of those custom libraries.
Can you please elaborate on intellectual property? If you paid a core to generate the model, or if you're using an FDA approved and presently prescribed medicine, does this require intellectual property agreements?
So IGNITE does not require for you to have all of your intellectual property in place, but because this is translational research and the reviewers as well as the program staff are forward thinking, we know what the next steps require. And we're trying to develop neurotherapeutics that will eventually make it into the clinic. So eventually you will need to show that you have freedom to operate, get a license. Sometimes people can change the formulation, the delivery, and there's some creative things people can do to get use patents. And we just really encourage you to work with your tech transfer office and have a plan for that, because when you move past IGNITE, there are stiffer requirements in place regarding intellectual property.
Can you clarify how PAR 21-122 is different than the BPN discovery phase? There seems to be overlapping goals. I'm wondering if I should apply to both or if that is frowned upon.
So you cannot have two applications under review at the same time at NIH that are not scientifically indistinct. So you would have whichever application, one of your applications will be withdrawn. So I would just say that for this particular question, I would really ask the PI to reach out to us. You can send us an email. We are NIH, but we are very accessible. Send us an email. We usually can meet within the month. And we can also bring people in on the team that would be able to answer your specific question. If you need people from BPN, well, Becky is actually a program manager in BPN as well. So we can have people on the team from multiple programs at NINDS to make sure we get you to the right place.
Great. And this is Becky. I'm going to add on top of that there's a tiny bit of overlap, but there's not a ton of overlap. So likely you're going to be better suited for one FOA or the other. And we're happy to work with you on figuring out which way to go.
Can you give advice or guidance on how to decide which mechanism, i.e. SBIR versus R01 versus IGNITE, to apply for? Will a better fit of other mechanisms be a factor during the review process?
So those all serve different types and stages of research. Again, the R01 mechanism is a little more basic research oriented, and this is early translational research studies. So this is NINDS money for IGNITE that you will have to compete for. As we said, the SBIR has the small advantage as in that you are competing for a smaller, you have fewer competitors for the SBIR money. That's a set aside amount of money that has to be spent. But you're welcome to come into IGNITE as a small business. Whether you go R01 or IGNITE will be determined by the stage of your research and where you are in your translational studies. And again, we're happy to meet with anyone to see what would be the best fit.
The next question is a bit project specific, so I would encourage you to reach out to Shardell or Becky individually. But if it's regarding whether you should submit a new submission or a resubmission. The next
I think
question,
that was actually easy to answer. Because you're applying to a different FOA, it would have to be a new submission, it cannot be a resubmission, is my understanding. And Becky, clarify if I'm incorrect in that interpretation.
Yes, that is correct. I'm trying to find which question we're talking about. Is that for applying to a, oh, okay, I see. Yes, that is right. Yeah, if it's a different FOA, you have to do a new application.
does PAR 21-122 include development of macromolecule drugs?
Yes, it
I'm not
does.
sure.
Both.
Yeah,
Oh,
I'm
go
not sure what
ahead,
the
Greg.
definition of macromolecule drugs are, but I think the answer is yes.
Yeah, both PAR 21, 122 and 124 support biologics as well as small molecules.
What is the pay line or success rate for IGNITE? I'm assuming for applying.
I'll take this one again. So we do not have a pay line for IGNITE. Things are paid based on what's in the summary statement, what the score is, whether or not the issues are fixable. Sometimes there's an issue that we can fix by adding a milestone, and that allows us to reach for something that we wouldn't have been able to reach for if it was pay line driven. So we don't have a hard pay line. Last time we did the analysis, which was before the pandemic, the success rate for IGNITE was pretty on par with an R01. But I'm going to caution you against what I think is maybe the reasoning behind this question. Lots of times we see people asking about pay line and success rates because they're going to pick the program that has the highest success rate. I would encourage you to rather pick the program that is more in tune or more aligned with what you're hoping to accomplish. Because if you are not meeting the goals of the program, it's not going to be likely to work out for you.
And then one more question, does there need to be substantial preliminary data before applying for PAR 21-122 or by extension, any of the other FOAs?
So I'm sorry, you're gonna have to excuse my voice. Yes, we allow preliminary data. It can be from your lab, it can be from the literature. It's important that you emphasize to reviewers that what you're doing there's rationale for it, that there's biological rationale that it's going to make a difference, be therapeutically relevant. And it's also important that you show that it's feasible to do what you want to do. And that's true for any of the FOAs. But beyond that, the preliminary data, like I said, could be from your lab or from the literature.
And that was the last question in the chat, but maybe we can wait a minute to see if there are any more.
Also, if you have questions, you can always reach out to us directly.
I think someone would like emphasis about preliminary data being from literature.
So you can build a strong scientific premise based on what has already been published, is what Becky meant.
Okay. So again, like Becky said, you can reach out to us directly. All of our contact information will be on the slides, and those will be sent out, like Anna mentioned in the chat.
Great. And Anna had one more slide.
Yes, I was going to say I'm going to turn it back over to Anna so she can present her slide.
All right, thank you so much. Thank you to Dr. Spriggs, Thomas, Ruth, and Hopp for a great session. Like we all mentioned, we'll send all registered attendees an email once the recording of this meeting that'll be on our website. And please join us for the next Open Stage webinar in June on HBCU alumni pathways to NIH, personal journeys and opportunities. I'll post the link to that open stage in the chat. Feel free to start registering as of now. And we're so excited to have you here and look forward to seeing you next time. Thanks.